Well, hey everybody, welcome back to biology. So we are in the middle of chapter 9, which is one of the more difficult chapters. Just so you know, if you're feeling that, it's true. And so what we've covered so far is DNA replication. And we said how, for the most part, DNA replication is amazing, being carried out by the enzyme DNA polymerase very accurately, considering the volume of information that DNA polymerase has to copy. It does a fine job, probably better than I would. But because of the volume of information, occasionally wrong bases are put into place, and that can lead to mutation. And mutations are errors. But it doesn't necessarily mean it's something that's going to be detrimental. In fact, if it wasn't for mutation, we would have less new traits, new things evolving. And so it is a source of newness. And so actually we have a lot to thank for mutation because it has led to speeding up evolution. So our next topic in this chapter has to do with point number 2, and that's protein synthesis. So not only does DNA need to copy itself, but we have to use that code of DNA. And so what is that code a code for? What do we use it for? DNA is used to make proteins. And as you can see, it's a multi-step process. But most living things are composed of proteins primarily, and so that's what the code is coding for in order for us to get the proteins that we have. It's coded on the DNA. So why do you have brown hair and no dimple? It has to do with the code that you have and the proteins that you made because of those codes. And so the DNA codes for proteins in that multi-step process. We got DNA, and to go from DNA to a working copy of that information, the messenger RNA, the process is called transcription, which is kind of like what you're doing now. You're writing, transcribing. So that's where the name comes from, transcription, or the copying of the information. And the second step is to go from copying that information to actually producing the protein, and that step's called translation, because we need to translate from one language of nucleotides to another language of amino acids. So we're going to learn how all life does this. So it's kind of a big agenda for ourselves today. It's like, how does life work? I think this is really one of the main components to understanding, how do you get from just that code that you got in your sperm and your egg to you sitting right there in your chair? How did that come about? It's from the DNA. And so we're going to learn how DNA does this. And DNA does this for all living things that have been discovered thus far. DNA, and in fact, it's the code, the A's, T's, C's, and G's, in the order that they're in, that's going to lead to a chinchilla versus a human. So we all have DNA, but our sequence is different. Actually, you and I, our sequence is different, but it's even more different between different species. So how does that then lead to these different forms of life? That's what I'm hoping to answer for us today. And this idea that DNA goes to RNA, goes to protein, was a huge breakthrough, and to show that the importance of this, that they figured out this process, they call it the central dogma. Central dogma of molecular biology is that DNA encodes for RNA, and that in turn encodes for protein. Any questions so far? Yes?
I was wondering, do all species have a double-stranded helix DNA?
Do all species have double-stranded helix DNA? Absolutely. Now, you remember we studied the genome of prokaryotes, and theirs is in a loop in prokaryotes, and then eukaryotes, it's in lines, but it's still all. Yep. Thanks. Okay, so I found this great overview picture, which I'd like to start with the big picture before we zoom into all the detail. So let's just understand this whole process that we're going to spend a lot of time talking about the particulars about. I already said DNA goes to RNA, goes to protein. So that's what we're seeing here. And I'm going to be discussing in a eukaryotic cell. And so we've got a cell here that has a nucleus. And then here's the DNA, which is cartoony here because it's way, 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 way longer. I hope you understand. But anyway, there's the double helix DNA. And so the first step, the transcription part, the writing, is the process of making the message, getting the message off of the DNA. So we're making messenger RNA here. So we need to get the information off of the DNA helix. So we'll see how that happens. And then we're basically getting the instructions, getting them copied off the DNA. And then the second step is taking the instructions and basically reading them or translating them and figuring out, well, what amino acid does this code for? And then gathering those amino acids and assembling them, and that forms a protein, because I'm sure you remember the building blocks of proteins are amino acids. So it's the instructions, get the instructions, then we follow the instructions. That's really what it is, summarized. And then those instructions will tell us how to make the protein. Good so far? Keep your questions coming, because it just makes it better for everybody. So here we've got an illustration of a chromosome. And as you know, it's all kind of twisted and doubled around and all very condensed around the nucleosomes and histones. But anyway, if you were able to loosen up a little bit of it, and you could see that it is indeed made out of that double helix. So DNA has information, and it has information in pieces. So a chromosome like that might have a number of genes on it. Let's just say an average of like 1,000. 1,000 genes on it. And so there's genes for different features, for dimples or hair color or height, or, depending on which chromosome, you'll find 1,000 different features on average on a chromosome. So the different pieces then of the chromosome where it codes for where it codes for proteins are called genes. So the gene codes for the proteins, and there's many of them on each chromosome. So for eukaryotes, it's super important to keep that DNA in good shape, because that's the code. So it's like if you messed it up in any way, then any future generation that inherited that mistake would carry it on. So it's best to try and keep it as mistake-free as possible. And so that's why the DNA itself doesn't get involved in making the proteins. The DNA itself is going to stay inside the nucleus. And so that's why we make a working copy. And so let me just lower the lights a bit here. And so we have the ability to make something like 25,000 proteins. And obviously, we don't need them all the time, all of them. So it really depends on what stage the organism is in, what's happening around it, what's happening in its environment. We don't need to, and we couldn't make all of them all the time. It depends on developmental stage. Like this is a nymph of, do you want to guess what this is when it grows up? Very good, dragonfly nymph. So the proteins the nymph makes is different than the adult. Like it's not making any wings right now. It doesn't need to. It's a nymph. And then a strawberry, actually, this is a picture of mold is what I wanted to show you. It's amazing to me that how quickly a strawberry can start getting broken down by a mold if you just let it be. And so the mold, which is, floating around in the air, just happened to land on a strawberry. And when it did, then it needs to start making all the enzymes to break down like the fructose sugar in that strawberry that it wasn't before it landed there. So it's like, things basically get activated and turn on in the right situation. And so it landed there and it's already breaking down the sugars. This is meant to illustrate some people that are sunning, and when you lay out in the sun like that, your body makes a protein to help protect you from UV radiation called melanin. Now, here in the classroom, you're not making it. Maybe there's people sitting by the window perhaps, but we're not making it here because it wouldn't make any sense to make it now. Why would you need to make melanin now? Go out into the outside, lay out in the sun, and then that protein starts getting triggered to start being made. So I'm just trying to demonstrate to you with different examples that at different times we have different needs, like umbilical cord. When's the last time you needed an umbilical cord? You've got a gene for it. You know how to make it. You've got proteins to make an umbilical cord. It would be really weird right now if you started working on an umbilical cord, wouldn't it? What do you need that for? You did at some point when you were an embryo, but you don't now. But I'm just trying to demonstrate that we have a lot of genes, and we don't need to turn them on all the time. And so this DNA to RNA to protein, I'm trying to get you guys to realize that we don't make all the proteins all the time, even though we have the genetic information for them. In fact, depending on what part of the body, different proteins are going to be on, being activated, being produced, and whereas other proteins would not be. Because, what you need inside of a muscle cell is very different from what you need inside of like a nerve cell or a bone cell or a liver cell. A liver cell, you might be producing, an enzyme that you wouldn't at all need in an egg, for example. So it's interesting, at least to me, that all of our cells have all the genetic information, all these, I'm giving you a rough estimate because we don't know exactly, but something like all the 25,000 approximate genes in each one of our cells. But each cell is only going to be making just a small handful of that, whatever's appropriate for that cell at that time. And the analogy I've heard is like an orchestra. Have you ever gone to like a concert before the concert started? And they're all warming up their instruments? How does that sound? It's like, ah! Because if everyone played all the instruments at the same time, it would be chaos. But that's not how symphonies go. So they go along a certain pattern. We can think of it like development, where sometimes some instruments will come on and other times they aren't. So protein synthesis is like that. And so it's controlled. It's controlled as if there was a conductor. It's controlled so some things come on at a certain time and then they're not appropriate anymore, and other things will come on. And so it makes sense to control this before you even start on protein synthesis. So if protein synthesis is transcription, translation, let's just control things at the transcription part. Why even go forward if we don't need it? So that's where where protein synthesis is controlled at transcription. My computer has a little message for me. Go ahead and plug it in. Any questions? Anyone so far? There, now it's happy. So then we have to think about, well, how is that done then? I mean, I'll tell you how you go from DNA to RNA to protein, but then why do we even proceed? We're not making umbilical cord. How do we not proceed there? So let's just jump now into the first part, which is transcription. So in transcription, we go from DNA to messenger RNA. And if we're talking about archaea or we're talking about bacteria, this is happening where everything happens, which is in the cytoplasm, because they don't have a nucleus, right? And if it is a eukaryote, this is happening in the nucleus because that's where the DNA is. So any process that involves DNA must be happening where DNA is located. And as I was trying to express in the last few slides, this is highly controlled. And so, there is an arrow going forward, but what makes it go forward? Well, it depends on which gene. Of all the 25,000 genes, which one is needed now, here? So which one is needed by this type of cell? Like what a muscle needs produced is different from what a liver needs. Muscle might need actin fibers for extending and contracting. Liver might need an enzyme. What developmental stage is the individual at? Are they an embryo? Are they adult, for example? What are the current conditions? Is the individual laying in the sun? Is the individual sleeping? Because there's different things that are made during the different day cycle. So RNA, we haven't talked too much about it, so I thought this would be a good chance to, let's compare and contrast RNA and DNA. We talked about DNA as being deoxyribonucleic acid, and the reason it has this long name is because deoxyribose is the sugar. So it's composed of, well, actually, just by way of review, DNA nucleotides are composed of the sugar and phosphate and a nitrogenous base. Right, there's those three components. And how DNA is put together is that the backbone is phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So if one compare that to a ladder, the sides of the ladder are the phosphate and the sugar. Now, the steps of the ladder, these are the nitrogenous bases, and they pair in a certain way. How does it go again? The nitrogenous base pairing? CG and AT. And so that's the DNA molecule. RNA has a role in making proteins, as I keep saying. How is it different? Well, for one thing, it's not a double helix. It's just, it doesn't even have to be helical. They just happen to show it this way. But it's single-stranded. This is double-stranded. This is single-stranded. It is made of nucleotides also. It's got a phosphate. It's got a sugar. The sugar's name is ribose. And it has nitrogenous base. The nitrogenous bases are, check it out, C, G, A, huh? No thymine. It's uracil. And so that must be important in the operation of it for bodies and cells to know what's DNA and what's RNA. Because if it's got uracil in it, it must be an RNA. So we'll talk about the base pairing and how that works, but you can think of it as uracil steps in and kind of does what you would expect thymine to do. So transcription starts with initiation. And for transcription to go forward, a whole bunch of, we're just going to generically call them factors, have to come into play and join here on the DNA molecule. So if all the factors aren't there in the proper arrangement, then nothing happens. And so I guess by default, our genes are in the off position. So you only go forward making the protein if every factor, and we'll see in a video how that works, every factor comes into play and they're in the right arrangement, and then it's go. but if they're not there. So that's how it's controlled. So it doesn't go forward unless all the factors are all lined up in the proper way. And then the enzyme that carries out transcription is called RNA polymerase, which looks a lot like something we just already talked about. What was the other polymerase we talked about? DNA polymerase, which is involved in which process? DNA replication. We're not replicating, we're transcribing. So instead of making more DNA, which is what DNA polymerase does, we've got RNA polymerase, which is making an RNA. We'll see, it's making an RNA. But it's similar in a sense. And so if all the transcription factors are there, then it can start. And so if they're not there, they're off. So it's more efficient that way, having everything off by default and then turning it on when there's triggers. Hey, we need some melanin. It's pretty sunny here. And then that will be triggered. They'll be signaled to get the production started. And so in order for RNA polymerase to work, this is a diagram from your book, we've got to get into that DNA. But the DNA, as you recall, is a double helix, and so it's basically hydrogen bonded together. So we've got to unwind it a bit, get in there in order to make the RNA. Now this is different than replication, because when it comes to making an RNA copy, we're not making an RNA copy of this strand and this strand. It'd be kind of like unnecessary to do that. When you copy DNA, yeah, it is important to make two full double helix, but that's not our point. We're just trying to make a single-stranded RNA. And so we only need to copy one strand. And so which strand? Again, there's all kinds of factors that would signal the polymerase which strand to go to, but we call it the template strand, the DNA template strand. An analogy that kind of works is, photographs and negatives. Actually, probably less and less these days. But remember when we used to use film? And there's photographs and there's negatives. There's one that makes sense, and there's one that makes you look really weird because everything's backwards. So it's kind of like that. It's like we don't need both because they both have the information. We just need the one that is appropriate for making this. We only need one strand, and so that's why we're only seeing bonding to one strand here. And this is also from your book. The template strand. So here's this haze here is represented as RNA polymerase. And so RNA polymerase will add nucleotides, which must be abundant. You have to have abundant nucleotides around for this to happen. It'll add the nucleotides in a certain way as long as the DNA is open there and it can continue to add them. It will add them in a certain way. Oh, I see, it's going this direction. And so the polymerase will just keep wiggling its way down and adding more nucleotides. So here's how it goes. Everywhere there in the DNA there is an A, it adds, you'd think T, right? But it's not. It's U, because we're talking about RNA. It's uracil. Everywhere on the DNA there's a T, it adds an A. Everywhere on the DNA there's a C, the RNA gets a G nucleotide. And everywhere there's a G, the RNA that's added is a C nucleotide. And so we're making a complementary copy. It's not an exact copy. You see, we're not adding a C where there's a C. So this can happen because of the attraction between nucleotides. What do we call that attraction? They want to be by each other. What's keeping them together? Hydrogen bonds. That's how this works, is by hydrogen bonding. It would make sense if you were going to design such a system that you do it more like a photocopier, where, if the recipe book says add milk, your thing would say add milk. But because it's a chemical thing going on and the nucleotides are being incorporated that are complementary, it actually comes out as a complementary copy of the template. Any questions right now about this process? So far we introduced the components here. We need the DNA because that's where the genes are. We need RNA polymerase because that's an enzyme that carries this out. We need to have a lot of RNA nucleotides because those are being added to one strand, the template strand. So we'll see this video that I took a still from. This is their representation of it. Here's the DNA, double-stranded DNA. It's double-stranded here and double-stranded here. The only place it's open up is actually kind of hidden because it's inside that enzyme, RNA polymerase. And so this is meant to be all the nucleotides, which, again, we get from our diet, the abundant RNA nucleotides in this case. And they're going to come in. You're going to see them channeling in and being added. And then what is made, it doesn't seem very long here, but let's just keep expanding this. Whoa! What's being made then is this enormously long messenger RNA. Because genes, on average, are something like 1,000 nucleotides long. So it's going to have to be really long. That's a really long copy. Okay, we've earned this. Let's watch this video.
What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
Isn't that amazing? How quickly that pairing can happen. So the only way this whole process, I'll just start it again, was going to go forward is that in this beginning, this kind of slow start.
What you are about to see is DNA's most extraordinary secret.
We needed to have a number of factors come in to that spot.
It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left.
So only when they all were in place, every last one, could it then proceed. So that's how transcription's controlled, by requiring transcription factors, and you don't go forward unless all the factors are there.
Process.
Any questions? Love your questions. Okay, so then it continues. It continues until the end of the gene. So, you know, it could be, like I said, around 1,000 nucleotides. But it will recognize, because of certain sequences that are present, that it's the end of the gene. I mean, otherwise it would keep going the entire chromosome, and that would be huge. But no, it just stops at the end of the gene. Because we just only have instructions to, we only want instructions to make one particular product right now. So when it reaches the end of the gene, then that messenger RNA, there it goes. Whoosh. And then the DNA is fully rewound. So it goes back to the state it was. The RNA polymerase hops off. Do you think it gets used again, would you imagine? Absolutely. Yep. Our cells are very efficient. So yeah, it can be involved in making another protein, RNA transcript. So I'm just restating this with a different figure. So when the end of the gene is reached, then the messenger RNA gets released, and the DNA gets rewound. So it's like nothing ever happened. That's our code. We don't want to mess it up. So let's put it back. Let's put the reserve book back in the library. We made our little transcript. Let's put the book back. And now we can use that messenger RNA. But first, there's a processing. We're going to process the messenger RNA before it leaves the nucleus and goes on to translation. When I first heard about this, I had to say I was like, I couldn't believe it because it didn't make a lot of sense to me. It's like, that seems like kind of a waste of materials and time and energy. But it's true. There is a processing, or there's removal. See how this is longer than this one? There's removal of pieces. Let's watch the video first, and then we'll talk about how that happens. So this is, the messenger has just been released, and it's going to be processed.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
Now, this is all wacky, right? Think about it. Let's say go to the library. There's a book that you really are interested in, maybe cooking some recipe from. You make the point of transcribing the information. And then before you leave the library, you take about half of those pages and you just throw them and then continue on. It's like, why did you bother transcribing it only not to use it? So that's kind of a more complicated question, and it has to do with the fact that that's how we have evolved. And sometimes unnecessary, or what seemingly unnecessary steps are taken, because in the end, we're not going to use those parts. But that's how it evolved, and it works, and so we continue on in that way. In some cases, we see that the pieces, the order that they're put together, make up different proteins. This is important in making antibodies. Anyway, perplexing. I just wanted to confirm if you felt like, what's the point in that? It does seem kind of strange. But anyway, it happens, and that's what I need you to know. It happens. So the parts that come out, they're called the introns. If you're wondering where they got the name intron, that means intervening sequence. So there's the intron pieces don't make the cut. I imagine that the nucleotides would be reused. You know, there's probably use for extra nucleotides, no doubt, but they're cut out. And then the exons are the pieces that get sealed together. So that's the coding regions. And to kind of just make it a little more durable when it gets out to the cell, typically there's a little cap made out of RNA nucleotides attached to one end, and then a little tail that's mainly made out of adenine nucleotides added to the other end to help it keep from being broken down so quickly, from being degraded. So this is what we made with the RNA polymerase. This is what we end up with. So we call that RNA processing. So which parts make it? The exons or the introns? Exons. That's what I'd like you to know. That introns get sliced out. So when we have all the exons put together and then we have a capped and tailed, we're ready to go for translation. But I felt like an obligation to let you all know that, you know, of our genomes, we've seen pictures of karyotypes, for example. How much of that DNA actually goes to this next step? How much of your DNA is actually translated? Turns out it's 1%. So it's almost like we're carrying all this baggage. And maybe you've done that. Have you ever gone on a trip and you packed way too much and you ended up just using like your bathing suit and your toothbrush and you brought all this other stuff? We're carrying all this other genetic material, and in actuality, we only actually use 1% of it. We are a product of our evolutionary past. There are some genes perhaps that were useful to us before we were human species, for example, and we still carry them. And then there's codes that will come in and kind of act like a computer virus. Maybe you know that too. If you're into computer science, you know that a lot of what's in your hard drive actually you don't even use either. It just kind of made its way there and you keep copying it. So anyway, we have a lot of material. Perhaps I just don't understand, or we don't understand the significance of it. Maybe it does play a role. Maybe it's important for us as source material as we evolve to new features. But anyway, just so you know, 1%, crazy, huh? Only 1% goes through translation, which is next. Transcription, just finishing up here. The outcome of transcription is the DNA gets rewound, messenger RNA gets processed, and the outcome is we have mRNA that's ready for translation. So are there any questions? Because we're this is like the midway through the central dogma. I don't want to go forward unless you feel pretty good about what's happened so far. So the process of transcription, where does it happen? In the nucleus. Is DNA involved? Yes. Both strands? No. The entire DNA molecule? No, just portions of it called genes. And are the genes made all the time? Are the proteins made all the time? No, only when they're needed. Great. Okay, I think you got it. All right, and then we're processing, and we don't have a protein yet, but we're halfway. So in order to do translation, and the reason it's called translation, it has a good reason for that too, is because what's the building blocks of DNA? DNA, nucleotides. What's the building blocks of RNA? RNA nucleotides. What's the building blocks of protein? Amino acids. That's different. So we're going from a language of A's and C's, etc., and we're going to another language of glycine and proline and valine and all these amino acids. So there's a translation that has to happen. And we're going to have a translator, too. Those lucky people that can speak 2 languages. That's what we'll see here. So translation follows the genetic code. And the genetic code is written in triplets. So just kind of a little preview figure here. We can see that if there's a code that has a TCT, AGA, like that, then that codes for serine. CAT codes for histidine, etc. Aspartate, proline. But it's in the point I want to make out is it's in threes. It's in threes. It's a 3-to-1 thing. Why threes? That's what this video does, I think, a really good job of explaining.
Once the DNA double helix had been discovered, the next big challenge was to work out how the 4 letters of DNA could code for each of the 20 amino acids that make protein. The first question was, how many DNA letters coded for each amino acid? If it was 1 DNA letter for 1 amino acid, then you could only code for a maximum of 4 amino acids. 2 letters in every possible combination could code for up to 16 amino acids. Still not enough. But 3 DNA letters provide more than enough combinations to code for all 20 amino acids. So 3 was the answer. It was a triplet code.
And here it is. This is one of the most famous charts ever worked out. And it was many, many, many scientists who put this together. This is not only the genetic code for you and your cells, but it's the genetic code for the chinchilla, for ivy, for little tiny worms, any living thing that you can eat, even viruses. This is the genetic code. And so it is, there is a table that has how many combinations on it? How many combinations of 3 letters are there? I'm counting more than 20. 4, 8, there's 64. Because there's 64 ways that those 4 nucleotides could come together. It's 4 raised to the 3rd. So, and it had to be more than 2, because like they said in the video, that would have been 16. So there's 64. And so it is a universal relationship between nucleotide, what's called codons. It's the first time I've used that word. Codon is the 3 nucleotide sequence, and how that corresponds to different amino acids. So I heard it earlier. How many amino acids are there? 20. All right, so what that means is that we have more than enough. And so, like right here, as an example, glycine. GGU, GGC, GGA, and GGG, they all code for glycine. So there's some redundancy. That's because we have 64 words, kind of, for 20 amino acids. It's kind of like the human language. How many ways can you say that guy's smart? He is also, you could say he's intelligent, sharp, what? Brainy, great. So you can think of these as like synonyms. So there, of the 64, 61 actually code for amino acids, have amino acids next to it. There's a special one. I wanted to be sure and show you. Here it is, AUG. Because it does two things. It codes for an amino acid called methionine, but it also says start. So it's like the start signal, too. And there's 3 codons that don't code for any amino acid, but they do code for a message. What message are they giving? Stop. And say stop to me in a codon way. Somebody tell me stop. Stop. UAG. So if I ever go over time on my lecture to say UAG, and I'll get it. I'll be like, oh, yeah. So those are 3 ways that you can say stop. 64 totally. And so a messenger RNA, this is the table for the messenger RNA. You might have noticed that it includes U's. Do you see them there? So you know this isn't a DNA table. I guess you could make a DNA table, but they traditionally don't. They usually show it as the messenger RNA table, codes for the messenger RNA. Like for that, we could translate that one up there, for example. So translation occurs in the cytoplasm. We're leaving the nucleus of eukaryotic cells, and the reason that it occurs in the cytoplasm is because it occurs at a structure called a ribosome, and you remember that ribosomes are found on the rough ER, and they're found free in the cytoplasm. Anyway, they're not in the nucleus. So this is like the workbench. So in order for us to construct this, and this is where all the amino acids are at, so in order to construct it, we've got to get out of the nucleus. The DNA is safe in the nucleus. We left it there. The messenger RNA emerges out of the nucleus. And the players are the messenger RNA, which, as you know, has been processed before it left. And it's going to be read in groups of 3 codons, 3 nucleotides. Then there's this funky-looking thing here called a tRNA, or transfer RNA. These are the ones that I say speak 2 languages. So they're going to be coming into play depending on the anticodon they possess. Yes, there are anticodons. And anticodons pair with codons, as we'll see. So they've got an important part right here, the anticodon, and then they're also going to be carrying an amino acid. So they speak both languages, in a way. And then ribosomes are involved, and the ribosome has 2 pieces to it. They've got 2 subunits, and they come together, and it's fed through. The ribosome is the message gets fed through the ribosome. So I just wanted to bring up these different components. So there's more than apparently one kind of RNA. There's the RNA that is the message that came from the nucleus, but you can see here there's also RNAs that are involved in translating that message too. This is just a different illustration of, here's a tRNA. And there's that part, the anticodon binding to the codon. This is a ribosome. It has 2 subunits. And I liken it to a workbench where translation happens. It happens inside the ribosome. So the actual translation itself is done through, it seemingly an enormously painstaking process, but then again, I can't invent anything better. But what happens is that there are a number, not just one kind, but many kinds, in fact, 61 different kinds of tRNA molecules that have, in the language of codons, an anticodon area, and they're carrying amino acids. So it's the translator. It's like, you say AUG, oh, I think I know what you need. You need a methionine. So this is the message. So just confirm that my figure is correct. If messenger RNA says AUG, what amino acid should be arriving? Do you see it up there? It just says met, but that is methionine. So let's do that for a few more, just so you get comfortable with the concept. Put a code here. This is the interactive part of our class. So here's another messenger RNA. Of course, it would be very long, longer than we would probably care to spend doing this. It would be maybe 1,000 long, but we'll just go for this many. Okay, if that's what the messenger says, then a tRNA is going to need to come and bring the right amino acid. So 2 questions for you is, what's the tRNA's anticodon, and what amino acid is it carrying? So go ahead and do the first one. I'll just draw the general structure here. Here's your tRNA. Put its anticodon down here, and then give it the correct amino acid. Okay, anticodon, everybody? Complementary base pairing. A? C, C. Well, I have a U there. It wouldn't stick. CCA. That'll stick because adenine from the RNA will stick to an adenine. Will stick to a uracil from another RNA. What's it carrying? It's not pro. What is this table of? This is a table of messenger RNA. Okay, so what is it carrying? Glycine. You could make, I guess you could make a table of anticodons, but that's not what the table is. It's a table of messenger RNA. So it's carrying glycine. Okay, you get the idea. Go ahead and do the next one. Anticodon is? GCG. Great. What amino acid is it carrying? Just give me the 3 letters. Arginine. Okay, let's see how fast we can do it. Next one. Anticodon. UAA. What amino acid? Which is the 3-letter abbreviation for isoleucine. Okay, next one, UGA. What do you say? When I say UGA, you say? Stop. Little trick there. Right. There is no tRNA that pairs with it because that's a stop. We need to have something indicate stop, and so that's how the stop is indicated. Okay, do you understand the role of tRNA? If tRNA is like going to a country where you don't speak the language with a friend who does. And you go to a restaurant and you're like, oh, I really want to have some of those rolls, and they can get it for you. If you asked for it, you'd end up with something you don't want, right? So it's the translator. It's able to speak both languages, and it's able to serve the needs. So let's see how then this all comes to play. We've talked about ribosomes, we've talked about messenger, we talked about tRNA. Let's see how it happens.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
So I'm going to break it down like initiation, elongation, termination again, just so you can. It's beginning, middle, and end. So let's talk about how did this start. This started because we had a message, process message, come out of the nucleus, and what happened as soon as it came out is this thing attached to it. What's this represent? Ribosome. And the ribosome has 2 subunits, and they come together. So the ribosomes are abundant and always there, ready for when that message comes out. And so what joins is that first tRNA, and it's always going to be the same one because they always begin with the start. It's just how it works out. The messenger RNAs all begin AUG. So it's always going to be a methionine that comes into play. So this is the message. This is the tRNA. Let me challenge you. What DNA code did we get to get all this started? Well, thank you for guessing, how we made this messenger RNA was from complementary base pairing. So it wasn't actually AUG. Yes. It would be the DNA that led to this mRNA, which led to this anticodon. I hope I'm not getting too complicated for you, but it's true. The DNA that led to this would be TACAAAGCT. Good for you, because it's DNA. So it's all done by complementary base pairing. Okay, so anyway, we've got the correct tRNA in place. We're ready to start. That's the initiation. The ribosome gets on. Well, actually, the message comes out. The ribosome gets on. The tRNA comes into place. So the next step I'm calling elongation. And that's where the messenger RNA will continue to be read. There's a place for 3 codons to fit inside of the ribosome. So this one's in place. So our next code here is UUU. And so we would need to have a tRNA that's complementary to UUU. So the tRNA would be AAA. And if you looked it up on the table, UU codes for phenylalanine, and that's what it would be bringing. So that's going to come into play and happening so much faster than I'm explaining it. I hope you understand. Dynamically fast, like in the video. So that comes into the right spot. And so the ribosome assists the correct tRNA coming into the right spot. There's many tRNAs. It's amazing to me how fast this happens because it only really is held by this attraction. So all the other tRNAs would be whirling around, but only the correct one that would actually stick and stay would be put into place. And then what then happens is that there's a third spot that says CGA in this particular example. And so we've got a tRNA that's got the code GCU that would be complementary. If you look up CGA on the table, you'd find it the arginine. So that'll come into place. But there's only space for 3 tRNAs. So if we want to start reading the rest of this, something's got to happen. What happens is that the very first tRNA, remember the one that was carrying methionine, that's actually going to leave. But before it leaves, it has to do something very important, is that it has to leave that amino acid. And so a growing chain is created here. They call it polypeptide chain, where the amino acid is attached onto the adjacent amino acid. Then it can take off. It did its job. It read what was needed. It brought the right amino acid. I'm out of here. What do you think happens to this if we were to follow the fate of this tRNA? Do you think it would be used again? There's a theme. You should get the idea. Yes, it's used again. So once that tRNA leaves, after it's brought its amino acid and attached it, it can leave, and then what it would do is find another methionine, and then it's ready to play again. So that initiator, that first one is released, so it's now out of the picture. And so we call that uncharged, and so when it picks up the amino acid, then it becomes charged, so it's ready to participate again. And then the ribosome wiggles down a little bit more. And so we can continue in reading this sequence, codon by codon, bringing the right amino acid. So one tRNA after another is going to come into the very limited area within the ribosome. And the tRNA that will stick and stay is the one that has an anticodon that is complementary to this codon. And it will bring an amino acid, and it will attach the amino acid. And then after it's attached it can leave. So just one by one, this is how proteins are built. One by one, the amino acids are brought by tRNAs. One by one, the tRNAs leave without the amino acid. And then they get recharged and participate again. And so what we get, if this happens enough, this is the messenger RNA. Here's the growing chain. Is we get a protein that's built. It continues on until what? Until it reaches stop. Give me an example of a stop. UGA, yeah. If it reaches UGA, then everything gets released. That means that's the end. We're done here. And so off comes the chain of amino acids, which, as you know, must be folded. If you remember from chapter way back, chemistry, they might be wound around in a helix or folded into sheets and then folded again. They might even have quaternary structures, other pieces that are put together, but they need to be folded so that folding happens according to what amino acids that chain is composed of and attractions that might occur between the different amino acids. So that gets folded. And the result here is that we are basically, we have a complete protein. that is released into the cell. I think it would be a good idea to just repeat that video now that we've talked through the process of translation. So let's just do this one more time.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA.
Do you see how there's
In this case,
3 spaces? There's 3 spaces for tRNAs. So if one's coming in, one's got to go. There's only 3 spaces.
the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
There's the protein floating off there. So I think it'd be good for us to compare DNA replication and protein synthesis, because there's a lot of similarities, but there's a lot of differences between them, and that would be a good way for us to review how the process goes. So I'm going to create a table, and you can join me as well. Is what? DNA replication. And we'll call this one protein synthesis. These are 2 activities that DNA is involved in. Be like a reporter. Go through the W questions. Where is DNA replication happening? Nucleus, great. At least if we're talking about eukaryotes. Where does protein synthesis happen? Tricky question. You should be asking me which part. Because it starts one place. It starts where? Yeah, we have transcription in the nucleus, and then we have translation in the cytoplasm. The best question of all, why? Cell division, exactly. That's when we need to replicate DNA. is in preparation for cell division. So every cell can get a full copy of DNA. We have the entire genome, basically all chromosomes. Undergo DNA replication. That's a project. That would be 6 feet of DNA in each cell. Okay, protein synthesis. Why? To produce proteins. So why would a cell want to produce proteins? Can it just be kind of generic? Do we need to produce all the proteins all the time? So we'll put here to produce proteins. What can I add? Needed, as needed. Exactly. You don't want every instrument in a symphony playing all the time. You want them to play it when they're needed. And so are we transcribing and translate? Are we transcribing the entire genome? No. So what are we transcribing? Genes, just the genes, which is a portion of the genome. So we transcribe genes. And then translate. So if I had to do it figuratively, this would be, I can't even do it. I can't even make it as long. I'd be like, okay, I'll see you guys in like 30 hours. That's how long the genome would be. Yeah, just keep going. Where here, if that's the entire genome, we're just doing a little portion of it. So just a fraction. So it's not as big a project, I hope you see. See, I'll put here, I can't think of a W question, but I want to ask you maybe which strands do we are involved in DNA replication? Which ones do we replicate? Maybe my question isn't so good. I was fishing for you guys to say both. You need to copy both strands. Because the goal is to go from one of those to two of those. And then for protein synthesis, do we do both strands? Just one strand. Do you remember what the name of that strand was? Begin with a T. Template. Good for you. Yeah, it was the template. So just one strand. The template strand. How about components? I guess that would be kind of a who question. What are the components involved? When we talk about it, what are the players that are involved and interact? For DNA replication, DNA polymerase. Are we good to go if we have those two? What are we building the DNA out of? Yes. What kind of nucleotides? There's two kinds. There's DNA nucleotides and RNA nucleotides. We're building a DNA molecule out of DNA nucleotides, right? Is RNA involved whatsoever in this process? No. Okay, the components. It's going to be a little bit different. We'll have to put, I'll just describe transcription and translation separately. For transcription, what are the components? Is DNA involved? You guys think so? Yes, because that's where the code is. Right. We need to go to the DNA inside the nucleus. That's where the genes are. One strand of them is going to be involved. And then what else is involved? Great. RNA polymerase. Anything else involved? What are we building this thing out of? Actually, maybe we need to put outcome, and that will get you there. Back up just a little bit. What is the outcome of DNA replication? What do we get in the end? 2 identical, if everything goes well, DNA molecules. What's the point of transcription? We're building an mRNA. That's the central dogma. Okay, so if we're building an mRNA, what are the pieces we're building it out of? RNA nucleotides. Excellent. RNA nucleotides. And for translation, what we need is that messenger RNA. We need ribosomes. We need tRNAs that have amino acids, with amino acids, and all kinds of factors that would help this process go along. And the result here is we're building a protein. Hopefully that chart will help you. So now that I've said all that, prokaryotes do it different. What's different? What's the major difference between a prokaryote and a eukaryote? A prokaryote lacks what? A nucleus. So in this process that we're talking about in protein synthesis, where you have something happen in the nucleus and then it goes to the cytoplasm, there is no point in waiting, because it doesn't need to go from the nucleus to the cytoplasm, does it? It's all there. It's all in the same place. And wait, they do not. Look at the amazing prokaryotes. So what you're seeing here, and they've actually seen EM pictures of this, is you're seeing the DNA strand. And on the DNA strand, we have the process of transcription occurring. And the mRNA is shown in blue. And so what's a little bit mind-boggling here is that, wait, we have got a messenger RNA here and here and here and here and here. That's right. They do transcription multiply. It would be like, let's go to the library and have that analogy where we have a book, except you're going with a team of friends, and you all are writing from the book. You're writing from different pages, but you're writing from the book. So we've got multiple transcription events happening. And what's this? What's on top of the mRNA? Ribosomes. What happens at a ribosome? The process of Translation. Translation is making a protein. Wait, each of the messenger RNAs has many ribosomes on them. So it's even before it's, okay, so we'll go back to our analogy. Here you are at the library copying down this information. Actually, a whole team of you are doing it. And then you're passing it to someone who's right behind you. Actually, someone behind you is overlooking your shoulder and beginning to cook the recipe that is written on that piece of paper. So I just want to say this so you can realize the impact of this, is that in prokaryotes, translation happens before transcription is done. In prokaryotes, transcription happens at multiple points. Think about the turnaround time. You know, for us, we're doing transcription and translation. Okay, we got that protein done. Think of how many proteins they can make simultaneously. But, you know, their life is much different than ours. Our generation time, about 25 years, would you say, between parent to offspring, more or less. Do you want to guess what prokaryote generation time is? Minutes. 20 minutes. So if your entire life was 20 minutes, you'd have to have a different tactic. Not that they're all that short, but they can be. And so, anyway, in prokaryotes, there's no editing, no introns. So the messenger RNA is basically all of it's translated. There's no removal of introns. And we have, because there's no nucleus, translation happens before transcription's even complete, and translation happens multiply on all of the messenger RNAs at one time. You're supposed to be impressed. Wow, how do they do it? Okay, thank you for your attention. I'd be glad to